My name is Velma, and I'm from the Luxembourg Center for Systems Biomedicine. In my presentation, I'll be talking about microbes and metabolites. The human body is the home to trillions of different microbes, and most of them are residing in the gut. I'll be focusing on bacteria, but there are also other types of microscopic organisms, such as fungi and archaea, as well as their viruses. Although microbes are best known as infectious organisms causing us trouble, most gut microbes are actually harmless. In fact, their activity is very important to the normal functioning of the gut and to the body in general, including the brain. The connection of gut microbes and the brain is known as the microbiome gut-brain axis, and this has become the focus of many researchers. As shown in this diagram, there are different ways that microbes can interact with the brain. Among the most important ones of these are molecules produced by microbes or microbial metabolites, for example, short-chain fatty acids. Other important pathways include hormones, the immune system, as well as the nervous system, particularly the vagus nerve. Knowing that gut microbes can influence the brain, an important next question is whether they could influence the development of Parkinson's disease. A key starting point to these discussions is the Braak hypothesis from the research group of Heiko Braak, who suggested in the early 2000s that the alpha-synuclein aggregation leading to Parkinson's disease could start outside of the brain, for example in the gut, and it would then spread to the brain via the vagus nerve. Experiments in animal models have shown that such spread is indeed possible. Since the complex communities of microbes in the gut are very difficult to study with traditional microbiology, such as culturing on a petri dish, we've only recently been able to start exploring the potential connection of disease and microbes in detail. The most common modern approach to studying gut microbes is based on collecting stool samples and using DNA sequencing of specific marker genes to tell what microbes are there. The first studies to use modern microbial ecology in Parkinson's research were published in 2015. One was led by Philip Scheprians in Helsinki, Finland, where I was working at the time, and there was also one from Japan and one from the United States. Since then, many more publications have explored the connections of microbes and Parkinson's. I recently counted as many as 30 of such studies with patient samples, and even more that use animal models or reanalyze and review past data. Due to technical differences and variation in human populations, the lists of microbes that are associated with Parkinson's are never identical across studies, but some patterns are starting to emerge. Here we have lists of some big bacteria that are often reported as increased or decreased in Parkinson's compared to non-Parkinsonian control subjects. Many of these decreased bacteria are producers of short-chain fatty acids, while the increased ones have been described as opportunistic pathogens that cause disease when they are in the wrong place at the wrong time. On the other hand, Akkermansia has also been linked to lean body weight, and Bifidobacterium and Lactobacillus are often used as probiotics. In other words, these are usually thought of as good bacteria that have beneficial effects. This can seem contradictory, but our studies so far tend to focus on large groups of bacteria that contain many species. So each one of these is a genus of bacteria, and there are many species within it, and even closely related species can have quite different features. So these lists of altered microbes provide us a starting point, but they don't really explain how microbes could be mechanistically linked to the disease. We won't be able to fully understand the connections of disease and microbes without knowing what defines our microbial community. The exact set of microbes that each person carries is influenced by many factors ranging from genetics to lifestyle. Last year, my previous group from Helsinki published a collaborative study with German researchers on people who have a high risk of developing Parkinson's in the future. We found that their gut microbes were influenced by factors such as smoking, exercise, and solvent exposure, which have been linked to Parkinson's risk in epidemiological studies. While we could then envision a sequence of events like this, where the environmental factors change the microbes then affect susceptible human cells to create disease, This still leaves open the question of mechanisms, and the whole picture will probably be more complicated. Medications offer a couple of different examples of relationships between environment, microbiome, and the host. Several research groups have shown that COMT inhibitors, particularly entacapone, can influence the gut microbial community. On the other hand, research has also shown that gut microbes can influence medications. So, for example, they can metabolize levodopa, which will affect its availability to the host. Maybe in the future, when we really understand these interactions better, we would be able to fine-tune medications based on the gut microbes. Aside from medications, microbes can also metabolize other molecules that their host is exposed to, for example, from diet or pesticides. So the end products of these metabolic pathways could be very relevant to disease development. One important group of microbial metabolites that I already named are short-chain fatty acids, or SCFAs. Microbes produce them by fermenting dietary fiber that we cannot use ourselves, and they are then providing us with an additional source of energy. SCFAs are thought to play an important part in gut-brain interactions, and animal studies have implicated them, for example, in influencing mood or behavior. A handful of studies have shown that Parkinson's patients have decreased levels of SCFAs. This goes hand in hand with the finding that many bacteria which are decreased in Parkinson's are SCFA producers. These figures here come from my latest publication from Helsinki, which we did together with Emory University from the US. And here we show that Roseberia, which is usually decreased in Parkinson's, is correlated to the levels of butyric acids, which is one of the most important SCFAs. On the other hand, Akkermansia here actually is almost entirely absent from patients who have high or controls who have high butyric acid levels. Akkermansia is important because it can degrade mucus, which is a component of the gut barrier, and it can then lead to an increase in gut permeability, which could lead to harmful molecules escaping the gut. Such a decrease in gut barrier function has been seen in Parkinson's. On the other hand, SCFAs help maintain a healthy gut barrier. In line with previous studies, we saw decreased levels of butyric acid in Parkinson's, although when we split this data into females and males, it was particularly prominent in males. On the other hand, women tended to have higher levels of stool calprotectin, which is a marker of inflammation. Inflammation is a much discussed topic in relation to Parkinson's pathogenesis and will be covered by other speakers in this meeting. It's also one of the most important connecting points between the microbiome and the brain and Parkinson's. So gut microbes exist in a constant interaction with the immune system. In a healthy state, there's a balance of keeping pathogens in check while leaving harmless microbes alone. When this balance is disturbed, it can lead to sustained inflammation that has unwanted consequences. In addition to other effects, SCFAs are anti-inflammatory. On the other end of the spectrum, we have, for example, lipopolysaccharides, or LPS, which are microbial membrane proteins that cause a strong immune response. When the gut barrier is disturbed, LPS can escape the gut, disturb the blood-brain barrier, and activate microglial cells in the brain. Another intriguing group of pro-inflammatory molecules are bacterial amyloids, which resemble human amyloids like alpha-synuclein. One example of them is curly, which is produced by bacteria such as E. coli and Salmonella to make biofilms. These are microbial community structures that help them stick together. Curly has actually been shown to increase alpha-synuclein aggregation in animal models, and this could quite directly connect it to Parkinson's pathogenesis. To summarize, our gut microbes, which are shaped by environmental factors, could be linked to the development of Parkinson's through an influence on alpha-synuclein aggregation, for example, via bacterial amyloids, or through inflammation related to increased pro-inflammatory molecules or decreased anti-inflammatory molecules. Both hypotheses are plausible and have some support, but neither has been definitively established. They're also not mutually exclusive, since inflammation itself influences alpha-synuclein aggregation, and bacterial amyloids are also pro-inflammatory. Well, one important thing to keep in mind is that at this stage, we still cannot completely rule out the opposite direction of relationship of disease and microbes. It could be that the microbial community changes that we see in Parkinson's are a result of the disease or the medications used to treat it. Some research does speak against this, for example, the fact that we can also see microbial community changes in early non-medicated Parkinson's disease and in REM sleep behavior disorder. It's also been suggested that maybe Parkinson's that starts in the gut is one subtype that is strongly linked to microbes, and it's just one type out of many. Now, whether the microbial community changes turn out to be a cause or a consequence of Parkinson's, modifying the microbial community could help alleviate the symptoms of the disease. There are already some promising early-stage results on this using either fecal microbiota transplantation, or FMT, which is replacing the microbial community with a healthier one, or dietary interventions, which we are actually currently studying in Luxembourg. To conclude, there's much we don't understand about the complex relationship of microbes and Parkinson's disease, but we do know that our invisible companions are very important. This is an active area of research, and we will definitely be hearing more about microbes and Parkinson's in the future.